Thank you very much. We are ready to start. I will say this series have been facilitated by Merit Healthcare Research and Innovation Landscape in Pakistan webinar series. And today we have honor and pleasure to present the faculty from Quaid-I-Azam University, who will be discussing the Pakistan clinical research landscape with a focus on Quaid-I-Azam University. So I am Umar Hayat, moderator for today. Just a brief background about myself. I'm a pharmaceutical development person. I have a position of VP of CMC and supply chain with Union Therapeutics. It's a Danish company based in Copenhagen. I have also founded HPC Consulting, but also launching clinical trials and animal studies in Pakistan. So the clinical research and the landscape in Pakistan really a lot of interest to me, but also to my clients in the US. So I will start with the faculty. I think Dr. Ansar, I'm going to leave to you, Dr. Ansar, to introduce yourself and dive into the presentation. My apologies, I have not prepared well today because I was switching my computers and I don't have the bios of each of you guys.
Thank you, Dr. Umar Hayat and Dr. Zia-ur-Rehman. Thank you very much for inviting Quaid-I-Azam University as well to present their research profile. I have attended some webinars on Al Khan University and they were really good. So I'm representing the Faculty of Biological Sciences, and in the Faculty of Biological Sciences, we have Department of Biochemistry. That was initially present as a Department of Biological Sciences, but later on in 2006, it was named as a Department of Biochemistry. So earlier we were a section in Department of Biological Sciences, but at the moment we are independent department, just like as I mentioned earlier, the 2006. So we have different faculty members which are involved in different research areas. Depending on their research priorities, they are running projects on different aspects. So I will introduce in the next few slides the faculty members and their research areas which they are involved in. So, but before sharing the faculty research profile, I would like to have a slight introduction of the department's academic program because we are mainly running the MPhil and PhD programs, but a few years back we introduced the BS Biochemistry as well because MSc Biochemistry is now fading out. So we have to start with the BS Biochemistry program as well. So like we just passed out the second batch this semester, this year. So that's not a very old program, just very recently introduced, but still we are having students with from different parts of the Pakistan, which are participating in biochemistry programs like BS, MPhil, and PhD. But the strength of our departments is basically the MPhil and PhD student, and they work a lot under different supervisors' lab. So you can see here the profile, the research areas where the department faculty members are working. One of the area which is established by our chairperson, the current chairperson, Dr. Murtaza, the signal transduction, the area of signal transduction, and she's doing a lot of work to establish the role of non-coding RNAs and miRNAs to explore their role in different types of diseases, especially the cardiac diseases. And then we have two different faculty members who are working in the area of cancer genetics, Dr. Mariam Anis and Dr. Nisa Sultan. They are working in the area of cancer genetics. One of them is exploring the cancer aspect at a genetic level. The other ones, they are Dr. Mariam is exploring the molecular mechanisms which are involved in different types of cancers. And then we have 3 faculty members which are involved in different aspects of plant molecular biology. But some of them, they are also involved in the natural compounds, their isolation and characterization from different types of plants. So based on a theme that maybe we can have new medicinal compounds from these natural resources. So Dr. Bushra Mirza, right now, she's acting as a vice chancellor in Lahore College Women University. Dr. Mina Shkil and Dr. Tahir Wahid, they are working on different aspects of plant molecular biology. Dr. Tahir Wahid also works in an area of DNA vaccines, and he has established a group to explore and prepare the vaccines in a way that they can be taken as like edible forms. So still a very early stage of his career and establishing new aspects, but he started with very good project that is in continuation with the European funding. So then we have Dr. Rizwan Alam, who is working in the area of cell biology, and he is studying the mitochondrial homeostasis and trying to explore the role of mitochondrial homeostasis in different types of diseases, especially platelet-related disorders and neutrophils-like aspects. So currently, recently he started new projects on neurological disorders as well, because mitochondria is one of the important machinery present inside the cell, and that is responsible for pathological conditions in many organs. So he's just establishing a new lab, but they have already started publishing some papers on the role of mitochondria and platelet-related disorders. Then comes the area where I am also involved in, and we are lucky to have Professor Emeritus Dr. Wasim Ahmed, who has established the area of human genetics at Quaid-I-Azam University in 97, 98. And then later on, I started the same aspect, but started working on different types of disorders. So later on, I will explore focus more on this area so that I can share the information there. And then we have Natural Product Research Group, which is partly done by Dr. Bushra Mirza as well. But in this case, Dr. Rashid Khan is also contributing in this area. So last year, we recruited a faculty member who started working on stem cells. So he's still establishing lab, but of course, he has expertise. He has worked in Korea as a postdoc and performed work there. So very briefly, I will have an overview of these the labs of the faculty members and the main themes of the projects they are carrying out, so that we can work out the future collaborative aspects if they can arise from these with these labs. So as I mentioned, the cell signaling therapeutic group that is working on 3 main aspects. So they're working on development of different animal models of different human diseases. So they mimic the human diseases in animal models, and then they try to explore the pathways which are responsible for those diseases. And then they are also trying to explore the therapeutic options against for the treatment required for the treatment of those diseases as well. And then they're also working on 2 aspects of the non-coding RNAs as well as the miRNAs. And they're trying to explore the role of these non-coding RNAs and miRNAs in human diseases, and especially the cardiovascular diseases. So they have a big grant that is currently on the non-coding RNAs. So as the other important group, where we have 2 faculty members working on this group. So they are working on the breast cancer and the head and neck cancer, as I already have explained while discussing the research profile of the faculty members related with the cancer genetic areas. So then come the cell biology group. So that's working on mitochondrial calcium homeostasis. And the main focus of this lab is that when this homeostasis is disturbed, how it affects the normal function of platelets, neutrophils. So that is how we can understand the platelet pathophysiology, cardiovascular diseases, and neurological disorders, which can be caused by the abnormalities of the different aspects of the mitochondria. So final aspect that is catered by the Department of Biochemistry and the Human Genetics, which I already mentioned that this is an area where my lab is also contributing. So in this research team, we focus on the identification of genes which are responsible for different types of inherited disorders. But because of some reasons of cousin-genous families and disorders which are autosomal recessive in nature, so we have developed a specific strategy to perform the whole genome genotyping and find homozygous regions which are shared by multiple members of the same family, and so that we can easily identify the disease-causing genes in those cases. So we are currently implying genotyping arrays, genome-wide genotyping arrays, and next-generation sequencing approaches to identify these genes or disease-causing genes in a big cohort of families with different types of phenotypes. I will introduce in the next slide. So for this purpose, we are having collaboration with many researchers across the world. So we have a very old collaboration with Baylor College of Medicine, Department of Human Genetics, and University of Washington as well for different next generation sequencing applications as well. So these are the disorders which we are working on since 2000. So even though I joined this department in 2007, but our department, as I mentioned, Professor Wasim has been there even before my joining. So we are studying disorders like hearing loss, hypertrichosis, microcephaly, intellectual disability, retinal dystrophies, polydactyly, skeletal disorders, and there are many others as well. But I'm just listing those ones which are major ones, and we have worked a lot on these disorders. I feel pleasure to state here that like if there are 100 genes known for hearing loss, about 30% to 40% of them, they have been identified by utilizing the families that were reported from Pakistan, and our group has played a significant role in this case. And the first gene for the hypertrichosis has been mapped by Dr. Wasim Ahmed as well. So that also belongs to the hair loss category that was there. So in all these groups, which I have mentioned here, we have identified multiple families with different genes, and those genes, they were reported first time from these Pakistani families. So this indicated that our group has used these families to identify new players which are responsible for different functions, and identification of these new players helped us to understand the normal mechanisms which are responsible for these functions. So I will share just one example to highlight our approach, which we use for these gene identification studies, so that it can give the audience a rough idea what type of workflow we use for these identifications. So I will introduce a case where the family, you can see here the 3 members, they are affected. There are 1 male and 2 females. So this family is suffering from intellectual disability. They also have ambulation delay, and they have delayed speech. So we collected samples from 6 members of this family. And then we performed the whole genome scan with microarrays, and then map these, perform the homozygosity mapping to find out the areas of the genomic regions which are homozygous in these 3 affected individuals. And you can see here in the panel C that the first 3 affected individuals, they are homozygous for a particular allele in extended regions of the genome. But the normal individuals, the healthy individuals, they are heterozygous. So this actually helped us to identify the genomic segment which probably carry the disease-causing genes. And then we use this second individual, which is circled by this red circle. So we subjected this individual for the whole exome sequencing, and the data was analyzed. And that analysis resulted in the identification of initially 75,000 variants, but further filtering resulted in the identification of 20 potential pathogenic variants. But 10 of them, they were present in the homozygous state. But when we compared these homozygous potential pathogenic variants, which of one of them they are present within the homozygous regions, which was already mapped in this family. So it turns out to be like only one pathogenic variant was present in there. And that variant was absent in the genomic databases like Genomad and dbSNP. So this indicates that the variant was rare and was also present in the homozygous region. So it makes our job easy to filter out those 10 potentially pathogenic variants. And we were now with one variant that is present in the TET3 gene, which is involved in the demethylation of the genomic DNA. And this variant segregates in the three members of the family. You can see here the variant was initially identified in only one affected individual through the exome sequencing, but later on it was through the Sanger sequencing, we confirmed that it is present in all three members which are affected in this family. So, when we share this data through the data sharing tools, so the variant matchers, and we came across that there are several groups which have identified mutation in this gene. And that resulted in a big paper in American Journal of Human Genetics, where we first time reported that TET3 is one of the genes that is involved in demethylation, because there are, before this gene, there are many genes which are identified, which are involved in the addition of the epigenetic marks. But this is one of the genes that actually removes those marks. So this is quite important for the regulation of gene expression. So this is one of the genes, but now we are getting the stories that there are other genes which are also identified with a similar function. So we anticipate that TET3 is responsible for the removal of the methylation signals by the conversion of methyl into the hydroxymethyl, and then later on the other enzymes, they remove that hydroxymethyl group. So this is the first step that is catalyzed by the TET3 enzyme. So the point of stressing this is that through these families, we can identify the mutations in genes which are involved in probably the new mechanisms, and those new mechanisms, they can help us to understand the disease better, and they can help us to understand how the normal brain functioning or maybe the nervous system development is carried out. So with this, I hope that I have been able to highlight the work that we carried out in our group, and if there are questions we can take up, or maybe towards the end we can talk about this.
No, thank you very much, Dr. Ansar. My apologies, I was switching between the devices. But I think we can move to the next speaker. We have to substitute actually Dr. Zia-ur-Rehman. He was busy with his some other commitments, and now we have our Dr. Tawfiq who will be presenting for him. Please, Dr. Tawfiq, you can go ahead and introduce yourself and then launch into the presentation. Thank you very much.
Thank you.
Thank you, Umar Hayat, and I am thankful to Uzair, also the host of this webinar. So let me first share this presentation and let me know. Is the presentation visible?
Yes.
Oh, okay. Thank you so much. Thank you, the Merit and APPNA organizers, to ask the Quaid-I-Azam University to host to showcase their research activities, particularly in the clinical areas. I'm here in place of Dr. Asanullah, who has to go to present the activities and the opportunities at the Department of Pharmacy, Quaid-I-Azam University. He is have some internet issues, and so he asked me. So it's as a stopgap arrangement. I'm here. So let's move. A Department of Pharmacy at Quaid-I-Azam University was started in 2011. It's a relatively younger department, and we are here to work in line with the vision of the Quaid-I-Azam University, which have the major is to provide the solution on the research-based solution to the national needs is the major theme in line with the today's webinar, I think. And the Quaid-I-Azam University aims to generate some resources or some thinkful on to knowledge-based economy. And in line with the vision of the Quaid-I-Azam University, the Department of Pharmacy at Quaid-I-Azam University also have some mission statements. And in line with the today's talk, I think that to strengthen the national economy through the discovery of the pharmaceutical, either from the synthetic route or either from the natural route, is our theme. And I think we can work together. And then we also aim to build a collaborative, diverse environment that will value the reward and innovation in productivity and the critical thinking. The department have some targets when we have incepted the departments, and we thought that we will come to the Pharmacy Institute of the Pakistan in first 2-3 positions within the 10 years. And then we also thought that we should move into the Asian ranking and the international ranking of the university in the subject of pharmacy and the pharmacology. And by the help of our research students, with the work of the faculty and then the leadership of the then chairman Professor Gul Majid, then Dr. Gul Shahnaz, and now Professor Dr. Sanulak, we are at the top of in Pakistan, first position in 2022, because we got first entry in 2020, and now we are at the first position above the University of Karachi, although it doesn't look nice to come above the University of Karachi and University of Punjab, our parent departments, but it's mathematics, and we are having in the position of the 151 to 200 slot in international ranking, QS international ranking. And this is only because of this major contribution is due to the faculty member. We have the 15 faculty member at present, and they are being supported with the hard work 75 almost at a time they are working 75 PhD and the 60 MPhil students, research scholars are working. And our faculty members, you will acknowledge that the 3 of the faculty members have H index more than 30 and 11 have more than 10 H index, and all of the PhD faculty members have obtained their PhD after 2010 or somewhere. Just introduction to the department. Department have been segmentized into the some sections: pharmacology, medicinal chemistry. Then we have the pharmacology, pharmaceutics, and the pharmacy practice. Today, I have divided the talk into the research areas wise. So first research area of the Department of Pharmacy, which is offering, is into the drug discovery and the development phase. And here we have the research team which are involved into the selection of the natural product and the natural product discovery, from plant material source selection to the extraction, isolation, and then further upgrading or the scaling to the yield. And then we have the pharmaceutical chemistry section segment where in silico studies and then structural elucidation part is done. And then these both natural products are from the synthetic source. The compounds or the extracts are being evaluated for the antioxidant, antimicrobial assays, enzymatic assay, and the toxicity assays. And these people, they are also performing some anti-inflammatory studies, analgesic studies, and then they have preclinical cohort of the hepatotoxicity, induced leukemia, and the wound vaccine models. And they are focusing in their areas of arthritis, diabetes mellitus, cancer, and depression. Remember, these people are working in the preclinical area and not the clinical. The human studies are not involved at this stage. And these people of the pharmaceutical chemistry and the natural drug discovery group have the facilities of the HPLC, microtome, fluorescence, microscopy, and so on. The persons who are involved and in the drug discovery are Professor Dr. Sanulak, whose main research areas are their specialty areas are natural product chemistry, drug discovery and development, and Dr. Humaira Fatma, assistant professor, who is also working this area. And in the phytochemistry and the medicinal chemistry, we had the services of Dr. Nasimullah, who died a little time ago, and we are going to have his substitutes very soon, I think. And the other research area related to the today's webinar is the pharmacological profiling. The people involved here, either the research team, faculty members, or their research team, they are using this from the source, either from the natural drug sources, either from the synthetic drugs, or the microorganism like the probiotics. They are working on the neuropharmacology, molecular pharmacology, or the immunomolecular pharmacology aspects. They are characterizing some Dr. Salman have a very good insight into the mechanistic approach regarding these natural and synthetic drugs. And they have some animal models technique. We have animal house, small animal facility at the current facility, and with the extension of the new department building, we have a dedicated animal house facility will be have within next year. Up till now, we are using the university animal house facility and small in-house facility in the current department of pharmacy. The pharmacology team included Dr. Salman Khan, Dr. Khalid Tipu, and Dr. Nadeem Irshad. Dr. Salman Khan is enlisted into top 200 scientists of the who are most cited scientists of the world. Then we have a section related to the pharmacy practice and the clinical research. I think these pharmaceutical aspects of the clinical research is looking by these sections, and the people here working, they assist the clinical trial. Some team of the clinical trial at the research organization, and they are working on some socio-administrative pharmacy research, some evidence generation, some observational studies they are doing. And here are list of the clinical trials where the team of the department of the pharmacy is involved, just like which have been registered. And these pharmacy practice people have established some collaboration not only inside the Pakistan, but also they are involved. They have generated a link into the Sinopac Pharmacoeconomics and Outcome Research Network. They have established a collaboration with the School of Pharmacy, University of Jakarta, and other health institute in the public sectors. Then another large number of the faculty members involved in the pharmaceutics they are working on the dosage form development. Either the dosage form is for the conventional drug molecules or for the natural products. And their main areas are the drug delivery systems, drug delivery techniques like liposomal techniques, solid lipid nanoparticles, transferosomes, polymeric nanoparticles, nanosuspensions, nanogels, or the hydrogels based drug delivery system they are fabricating. They are loading drug, and then they are making some characterization technique for the preclinical studies. The drug development and the formulation group have the facilities of the zetasizer, rheometers, probes indicators, and France diffusion cells for there, and they have the services of the HPLC, DSC, and so on. They not only use their in-house facilities, but we have also good collaboration with Department of Chemistry, Quaid-I-Azam University, and National Center for the Physics, NCP. The drug development or drug delivery groups have the number of the researchers. Dr. Gulshanaz was leading this whole group. So she is nowadays moved to the USA for the postdoc, and hopefully she will come back next August, I think. Then we have Dr. Hussain, mostly interested into the IBS and the nanoparticle-based drug delivery approaches to the bowel syndrome. Dr. Naveed, he is working on the nanotherapeutics and the nanomedicine diagnostic approaches, and Dr. Fayatullah and me are working on the transtrangle and the topical formulation for the conventional and the natural products. And we have the services of Dr. Fakhruddin, who have some patents before coming to here with his Korean counterparts. And when he arrived here, he worked on those patents, and he have generated and one more patent from the university Department of Pharmacy at the university in the drug delivery area. Then on the point of view of the commercialization, the natural product research group have come up from some product, QLAC and QCEF. They have worked with the ORIC, and they have commercialized one of the two products, these were. And at the same time, department people have applied some for patents, two or I think three patents they have been awarded, and rest of the applications are under process. And at Department of Pharmacy, we have a collaboration with Islamic University Bahawalpur Department of Pharmacy, Higher Education Commission. Then LAMS, we have a collaboration with the researcher there and the University of Queensland. The funding at public sector universities are always the problems, and so some of the faculty members have been successful to get funding from the NRV force or the IFS or the Pakistan Academy of Science and Pakistan Science Foundation, UNDP. In future, or from the APPNA perspective, we need some collaboration from the International Pharmacy Institute so that we can have some prerequisite degree program for the USA, where they offer 2-year program. They have to pass the 2-year program in the basic sciences. So we are planning to have some collaboration so that we can prepare the Pakistani students to have admission in the USA universities in the pharmacy concept. And then we are thinking, or we are working on the nutraceuticals and the cosmeceutical, apart from the pharmaceuticals approach regarding the product development with the collaboration of the local industry. And then we also want to have some foreign faculty members, either they are Pakistani origin or from the other nationality, and they can spare sometimes online so that we can manage some education of our undergrad, postgrad, or the research students, or some research collaborations. So if you need a contact detail, Chair Pharmacy or Dr. Ahsanullah may be contacted. We have a PC1 of the new building, and hopefully by the start of the next year, or by the mid of the next year, we will get that building. The building at the rightmost panel is shown is going to be completed soon. And thank you so much for sparing time and giving me some opportunity to Dr. Omariya. Thank you.
Thank you very much, Dr. Tawfiq. Do we want to go to Dr. Rabaab if he has a presentation ready? Dr. Rabaab, do you want to present?
Yes, I basically, to be honest, I'm very sorry. I'm having lots of issues with this machine today. Maybe it's a bit overused because I have lots of meetings today, but I have a very short presentation. I will just share this with you guys in a second.
No worry. I was thinking I was the only one who was having the problems.
No, I had unexpectedly, I had a number of meetings today, and I'm just very sorry about that. And in this whole process, I've kind of seemed to have lost the actual one that I wanted to present, but I've just pulled up this thing because I would like to just give a brief, and I'll skip through lots of the materials as well. I don't want to take too much time, just to give a brief what exactly we do.
Perfect.
Just hold on.
Go ahead.
Sure. I'm going to. Okay, I should share. Yeah, sure. I will only share my presentation actually. Yeah, that's fine. So. Is it?
It's
No,
there. Thank you.
do you see that?
Yeah.
Oh, okay, great. I was a bit unsure of what exactly I should speak about, but I'll just go briefly. My name is Rabaab Zahra. I work in the Department of Microbiology in Quaid-I-Azam University. I work primarily on an area which is antimicrobial resistance, which is, I think, is pretty much known to lots of people, even who are not in the field, because it's of public interest. I have put up the talk as gaps and challenges, but the time that we have, it's not, of course, enough to go through everything, but I will just discuss briefly what my perspective in terms of this. So if we think of the problem of antimicrobial resistance in AMR, in particularly from our perspective, pretty much everyone knows that there is a significant burden of resistant bacteria overall. So these resistant bacteria, they are from the gram-negative, gram-positive, they cause number of infections, and then we have number of categories of these resistant bacteria, and World Health Organization has come up with now prioritizing these pathogens. So particularly I've put some of the names here, like ESBLs, NTM, like CREs. I apologize if these are a bit unfamiliar to some of the audience, but particularly anyone who works with microbiology and with the AMR, these are familiar terms for them. The problem with AMR is that it leads to significant mortality and morbidity and leaves the clinicians with very limited options for treating the infections. So I particularly would briefly describe what exactly in the Department of Microbiology we've been doing this. So we are very focused looking at this burden of the resistant bacteria in neonatal infections. So okay, I'll pretty much skip this, but this is a bit old data, but this still holds true till this day, because according to the child mortality report of UNICEF, there are a number of countries around the globe who are struggling to meet the challenge to reduce the neonatal mortality. Pakistan has made significant progress if we observe this over the year, but still we haven't met the target. So in other words, the global challenge or the sustainable development goals to achieve the neonatal mortality ratio, we're still lagging behind. And the
Dr. Rabaab, are you moving your slides? Because we are seeing only first slide.
Yes, I'm moving. I'm at the slide number 4, actually. I'm sorry. I'm moving it. Don't you see the child mortality report UNICEF slide? I'm at the slide.
Dr. Zed, do you see the slides update as Dr. Zahra is moving through?
No, we cannot see. We can just see the first slide.
Yes,
I'm sorry. I'm actually at slide number 4.
Do you want to do the slideshow and then probably?
I did the slideshow. It is an slideshow. So.
Do you want to stop and share again? That's one way to work.
Okay. I'll do the share again. Okay. This time I'll just move the slides. Do you guys see the first slide?
We do this first slide, yes.
Okay, and if I do a slide,
okay.
Yeah, if I do a slideshow.
Yeah, do the slideshow, please.
So, which slide do you guys see? It should say number 4.
Still see the first one.
Still see the first one. Okay, there's some problem then, and I'm really sorry. I don't know why is this happening. Unfortunately, I mostly use Teams. I'm not very familiar with the Zoom sharing, although I do so many meetings on Zoom as well, but somehow I'm not really.
Okay, now you are on the 4.
Do you guys see the slides?
Yes, we do the number 4. Go please to the, yeah.
You see the number 4?
We do the, yes. The number 4. Yes.
Does it work now?
Yes.
Yeah.
It's working. Okay, I'll just, because of the interest of time, I'll just move around. So simply Pakistan is struggling to meet the challenge of the neonatal mortality and morbidity. And this slide simply is the slide number 5. It simply shows that most of the neonatal deaths happen in the first week of life. So essentially, if you see this map here, this is a map which has marked images. So this is we at Quaid-I-Azam University, we were part of the consortium. We did this study, Barnard's, which is the burden of antimicrobial resistance and antibiotic resistant bacteria neonates in developing societies. So it was a partnership of 7 countries. It's Ethiopia, India, Bangladesh, and UK with the main site. And we also had Rwanda and South Africa. We did this model where we tried to understand the reasons for the neonatal sepsis and the factors which are responsible or contributing to this problem and what is the outcome. So essentially, we looked at the maternal risk factors, environmental factors, neonatal factors, and then the downstream effect, what happens with the survival and if there's a mortality. So in between, what's the clinical picture and the microbial data. This study was mainly done at that one public hospital in Islamabad, which is PIMS. It's quite familiar to everyone. PIMS has a children's hospital and the mother-child health center. So we catered both of these facilities for a period of 3 years. These are some of the findings, but I'm not going to go into the details because pretty much most all of the data of the study is now published. So we found number of resistant bacteria, and we also found the outbreak of particular bacteria. These graphs simply show some of the variability of the different species of bacteria like Klebsiella and Serratia and their resistance profile. And it wasn't a surprise for us. We already knew that we will be finding resistant bacteria, but I think a bit was that we didn't think that it will be too significant. Even in the neonates, they were very, very resistant bacteria. And we tried to look at some of their outcomes and what was the positive culture samples, what was the impact on their overall. So there was a 20% in the study population where we found they actually just couldn't make up to this. And even those with, there was a discharge or the patients who were fine and they were discharged from the hospital, some of had lots of comorbidities as well. And it is also well known in the neonatal health that the comorbidities along with an infection makes it very difficult for a clinician to deal with the particular case. So as I mentioned, I will not really be too much details on this. This is the study we did again at Panorama study. It was on the bacterial, the sepsis patients, and it was actually done in 22 countries, a big cohort study. And again, just highlighting the fact that the resistant bacteria are problem for. It's actually a problem for overall the globe, but particularly these bacteria are a problem for middle-income countries and low-middle-income countries. And then some of these papers are related to our Barnard study. So basically all the data is available if anybody is interested to understand what exactly parameters were in this study. So this is all available on the site. And I just want to mention that along with the neonatal work, this is something that we have started doing. We are also looking at this problem in the environment. And this, if we just look at this review, every AMR talk has a reference of this review. This was actually written by an economist, Jim O'Neill, in 2016. And this was his review, which actually kind of awakened the world. And that's based on this, that the British government decided to put up lots of money into the AMR field because there is a prediction, and the prediction is not far from being true, that if we do not tackle the problem of AMR in today's time, we actually will be running into this like we just all observed a pandemic. So it's a silent pandemic. So by the year 2050, you will have at least more than 3 million people will be dying because of this. And the one more striking feature is that among all those 3 million deaths, you'll have one particular bacteria, and that bacteria is the one which is your drug-resistant E. coli. And E. coli is a very common bacteria. All of us carry it because it's our gut bacteria. And we tried to look into this, said, so this is basically going a bit into the genetics and ecology and evolution of bacteria. So along with these neonatal infections and the clinical burden, we realize it is very important to understand the problem, but to understand the problem, you need to connect the dots. So our environments are linked, our clinical and non-clinical environments are linked. So we did this study. So I think reference of this study was here. This is a very, it's not just that our lab did it. Basically, it's a, I would say, kind of a struggling paper, because before this study, most of us were under the impression that, oh, if Bangladesh has a particular E. coli type, probably Pakistan will also have the same, but this is not true. So we did have a very drastic difference of the sequence types of E. coli, and as compared to the different other areas of Europe and even South Asia, we had very different E. coli STs. So simply my lab works on different genetic and ecological aspects of these bacteria as well. So essentially in Pakistan, it's now Pakistan government has taken up it as a challenge because of the guidelines of WHO. So we have a national action plan. It is in place. We have lots of funds coming from through Fleming Fund to Pakistan. Our NIH has a reference lab. Then there are Sentinel sites. And so much work is being done to cover this problem. And of course, Pakistan has been now on this verge that the Ministry of Health, along with that, they're doing this One Health approach. And there's a nice coordination between all sectors of the health and non-health sectors to tackle the problem. So my research group at Quaid-I-Azam University is trying to do their part. I will simply just say thank you so much to all of you. And I just wanted to acknowledge some of the funding organizations who have helped us to find some of the answers to the questions. And I'll be here. I'm sorry for some of the problems, but I hope I conveyed some of the areas that we work on. If there are any questions, I can take later on whenever you do a QA.
No worries, Rabaab. That was literally very important topic you were touching upon, and unfortunately, we shared the pain, your slides and technical difficulties you were having. But for the audience, we will share the Dr. Zahra's slides for everyone, so that please share with us your email, and you will have the presentation slides. So with that, thank you very much to all presenters. We will go around the panelists. I think we have Dr. Azhar. Dr. Azhar, you want to say a few comments? Then we will ask the audience if they have any questions, they can raise the hand. We will unmute and you can ask the question directly to the presenter, or you can type in the chat box and we will pull one by one and ask the question to the presenters. Thank you. To you, Dr. Zahra.
Thank you, Umar. So first of all, we are extremely grateful to all the valuable academicians and the participants for such a wonderful presentation. Very well organized. I think everybody, they complete in the assigned time. So thank you very much. And it's an eye-opener. So much impressive work is going on in Quaid-I-Azam University. So that's why that is the premier university in sciences in the public sector in Pakistan. I remained as a member National Commission on Biotechnology for a good length of time, and at the same time, I'm so happy and excited to share with Rabaab that I was the first General Secretary of Biosafety Association in Pakistan when they were thinking to start the work on antimicrobial resistance. So I think I'm so happy that they are flourishing and a lot of activity is going on. And at that time, you know, that was the transition between Professor Amir Ikram, and now NIH is the place really to look to. I have a few comment and question. The general question is, first of all, is to Professor Dr. Muhammad Ansar. I could see, you know, a lot of activities going on in the field of genetics. And of course, Pakistan is the place to look for and probe and exploit the opportunities in the basic research in the human genetics. And I think the activity started way back in 70s. My question is that I could see there is a lot of linkages with the global institutions. But I don't know the hurdles, but I do see a disconnect between the local uh, clinical fraternity. I mean, there's a lot of work going on at different places. So is there any special network or a body, which includes all the people who are working in different aspects of genetic diseases in Pakistan? So this question is to Dr. Muhammad Ansar, please, if you would like to respond to.
Yeah. Thank you, Dr. Zahra. So basically, I agree with you that the work started very earlier, and Dr. Qasim Mehdi was, under the umbrella of Kerala Labs. So he
Yes.
set up the genetic studies first, and then followed by Quaid-I-Azam University, where Dr. Wasim actually carried that work. So unfortunately, the problem in Pakistan is that we have a like disconnect between clinician and universities, academicians in the universities. So, like I mentioned in the presentation, that we have a cohort of 4,000 families, but only 2 or 4 families they are recruited through the involvement of the clinicians. Rest we have searched ourselves. We visit those remote areas and we go village to village and then find out the individuals with multiple individuals in the family having a particular condition. So that's why there's actually 2 reasons for this. Maybe number one, that the individuals with the inherited disorders, they are told by the clinicians that they have no treatment for that. So that's why they're not following in the clinics, and as a result, in the when the future child is born with the same disease, they do not visit the clinic. So as a result, we have the first task, to characterize those patients, and then we can work up in the larger extent. Like right now, there are some labs which are establishing to start genetic testing in Pakistan. So we have a genetics lab established in Lahore recently, and few groups they are planning to set up additional labs as well. So, but of course, if we want to offer services, coordinated services, we need to know the profile first. So our part is to characterize the profile, and then the stage will be set to perform diagnostic services in the future as well.
Great. Perfect. So, I would like to add, as of today, there is no clinical clear guideline about the screening or the genetic diseases to the pregnant women and even to the lead institutions like Shifa International Hospital or Doctors Hospital, National Hospital, or in Karachi in Aga Khan. So these aspects are missing. So we need to sensitize and educate the clinician, use as a screening place that they can refer, and they should collaborate with the researcher. I think some work is going on by General Sohaib, Brigadier Nusrat Salamat. And I remember we had a couple of webinars in the past. I will share with you the link
Okay.
about the genetic. I think we have the 3 such webinars. And
Mm.
some people, they had shown interest from the international community to go for some joint activities. I will get you connected with them because we need to give really importance to these things. Generally the perception from the patient community is that the researcher, they are doing the research, it's going to be published internationally. But at the end of the day, at the receiving end, they are not getting like counseling, like follow up, like what should they be doing. So
Yeah.
these are the challenges locally, and they were highlighted many times in the past. So, and there was some work which was going on by the Center for Advancement in Molecular Biology, and it was published with the US collaborative centers in the past by Sheikh family. So I
Yeah.
still
Yeah.
remember there was some work which was going on. So
Yeah.
that was question to you, and we will be in touch with you and we'll get you connected with that. Moving
Mm.
on to Dr. Tawfiq. So congratulations to have the such a wonderful pharmaceutical sciences department, which is though new but creating a real impact, and stood number one. So that's very, very impressive, and we look forward to provide you the support. So your presentation is extremely concise, well focused, and highlight the importance, and at the end of the day what we are looking out of it. So that's very impressive, and we will take this initiative to get you connected in both the resources in the natural health product as well as the pharmaceutical delivery system. So yesterday again, I was in a meeting with some of the people, the global people who are looking into the drug delivery system, and I am also in contact with the life sciences or the pharmaceutical industry back in Pakistan, which is growing. And we need to bridge the activity between the uh, training and the teaching and the education and the industry. You know, there are different routes. So we will definitely find a suitable match for you to provide you the support in getting your student to the US. But at the same time, I do have a plan to visit Pakistan soon after, like in about few days. So I'll be happy to meet you and just to facilitate the bridge between the industry requirement as well as the academician, the role, the disconnect, and basically, you know, how to bridge that activity. And probably Quaid-I-Azam University could be the first one to take the initiative in this regard because that is the missing link. So stands true for even the North American market. So we will be very happy and accept my congratulations for such a wonderful presentation, very well focused and leading to somewhere. The last speaker, Dr. Rabaab Zahra, I think that's a very impressive and that's a big challenge to Pakistan, and it's a very complex activity about the antimicrobial resistance. As we all know, it's a multidisciplinary approach. It's not limited to just the clinical side or the basic sciences side. It's a very complex. It also relates to the medical practice as well. Like in developed countries, be it UK or US or any other country, there are defined clinical guidelines, and all the clinicians, they follow the clinical guideline by the use of chemotherapeutic and antibiotics and stuff like that. But as you know, in Pakistan, even you can get the antibiotic from the medical store, and with all the challenges about the discontinuity selection, so microbiogram and those things are very limited. Unfortunately, as of today, the basic microbiology is strong enough in Pakistan. However, the clinical microbiology, the application is very limited. If I could remember, like 10 years down the road, 10 or 15 years, there were hardly any proper hardware available to the leading institute. I mean, the complete automation in microbiology in Aga Khan University, Shifa, Doctors Hospital, National, just to name a few of them. And now this discipline is going to take root in Pakistan with the introduction of good quality automation in microbiology and identification as well as in the sensitivity and all those things. Some leading solutions are available. And in this, even in the regional picture, I'm talking about the clinical microbiology, I definitely see that there is a gap between our neighboring countries. We are far behind. But from the basic size, the basics and microbiology, I think that's very good. And then there are other applications as well as the industrial microbiology, and you know the spectrum is quite wide. So that's great. There was a question in the QA box about the animal facility available with the Quaid-I-Azam University. So anyone of you would like to share what type of animal facilities do you have, and what type of animals you have, and how large is that, and what is the population? You know, just a brief. Somebody mentioned in one of the slides about
I think Dr. Tawfiq this is a question actually. I would like also to know what are the animal models, and also what are the animal models for certain therapeutic areas which
Yeah,
are being studied at Kazim
yeah,
University.
yeah. Dr. Tawfiq, please.
Okay, thank you, Sayyid Uzair and Umariyat, for nice introduction and about our Quaid-I-Azam University regarding animal house. Small animals like mice and rats, and large animals, the Department of Animal Sciences or Zoology are looking into working on some monkeys, and Dr. Shahab was working on monkeys previously. I don't know nowadays, but they have a large animal facility there under the custody of the Animal Science Department. Department of Pharmacy under the PC1 new building, we have 4 large rooms, and we intend to have focusing on the small animals like mice and rats, and to some extent rabbits, but we don't have the facility for larger animals like monkeys or goats at that dedicated pharmacy facility. And the researcher working in the pharmacology and in the natural science, they are working some animal model like diabetes, some anti-inflammatory models, or some depression models on the small animals also, not the large animals.
So I think that can be a wonderful opportunity, Dr. Azhar and Dr. Tafiq, because I can see a lot of pharmaceutical companies who would be very much interested to screen their molecules or formulations in animal models, and especially for large animals like the dogs or monkeys, as we say always, the PK scale-up, they become very, very important. Not only important because they want to see how the translation of the pharmacokinetics goes from the small mouse studies all the way to the humans, but also the large molecules are very expensive over here. So I think the Pakistan can present like as a destination for animal studies. The one thing I would like to mention and ask Dr. Tafiq, I see the lot of dispersed expertise and capabilities. Even in one of your slides, I saw the structure elucidation. I mean, in your discovery group. So I think that's also some of the capabilities we need to have, and along with the synthesis of the library of the molecules. So I think those are the areas. For some reasons, I think even this has been always the question and point we have been making. Like there should be a consortium, so the different departments with their existing capabilities can collaborate, and alone probably they are not complete, but as a together they may be complete. The other option is always like a pick one center and put all the possible the equipments and the whatever we need for that particular activity, if it can be the discovery or structure elucidation, or if it can be the animal studies. Like we can have even a central vivarium. I don't see why we cannot have it. So for structure elucidation, how far you guys can go? I mean, do you guys have the NMRs, the MS, LCMS? Can you elaborate on that one?
Thank you, Umar Hayat Saab. Regarding the pharmaceutical chemistry section of the Department of Pharmacy, this section is working in a close collaboration with the Department of Pharmacy, well established at the Quaid-I-Azam University. So for the structural elucidation, they are getting sample analyzed from the NMR, a small NMR we have at Quaid-I-Azam University. But for the small proton NMR we can do here. But for the extensive work, we send sample to the HJ Karachi. Institute, Karachi, Dr. Tauram and Dr. Qual-Jofri. And they are already working in the collaboration with the facility, like this crystallography. We don't have a crystallography here, but the workers either in the natural product chemistry or the phytochemistry, they send sample to, or they have a collaboration with the University of Sargodha, where they have a good crystallographer. I don't remember the name, but they used to have a work in collaboration with him. So you are absolutely right that not one department can have all the things, or the one institute. So we need to establish, in my opinion, one state-of-art lab, not in one university, but that should have the collaboration, like this scanning electron microscopy or the transmission electron microscopy. Not every institute can afford to operate and run that expensive equipment. But there should be some protocols and the procedure for the facilitation of the researchers or the students, because there is a large use at University of Peshawar and other institute where the people have the scanning electron microscope. So I will request you people that if you can facilitate this type of consortium where the researcher can be facilitated through the small token money of the research from their research fund. So it will be a good help to the Pakistan, in my opinion.
So after we need to pull probably the industry. Can you imagine all pharmaceutical companies in Pakistan? They cannot even synthesize in-house the reference standard. All they need is basically a lab to synthesize. I mean, we not produce at the large scale normally the reference standard, and then the structure elucidation and the complete analysis of the compound. So I think if this can be done in probably collaboration with industry, that may help not only the research institutes or academic institutes, but also to the industry.
Dr. Umar, just two housekeeping. Number one is there is going to be an overlapping webinar going to start shortly. So we need to be move on very fast. And secondly, now you have a trip plan for Pakistan. I'll request you, please keep in your agenda item, get connected with Dr. Tawfiq and visit Quaid-I-Azam University, the pharmaceutical sciences, and see what can we do as an individual or as a group or at a big platform level. So that is going to be my humble request to you. And with this, I think we have a message. So please, I'll request you to move towards closing the session, please.
Thank you very much, Dr. Azhar. So one announcement or request we have, and I extend this request to all audience as well as to the presenters. We always looking for the people who can participate on the panels, on the merit. It doesn't necessarily need to be always from US or Canada. We encourage that people should participate from Pakistan as well. And if you have the interest as a moderator, as a panelist, or as a speaker, please share the information with the APPNA merit email, and you will have in when you receive the invitation. With that, I think we thank to our speakers. Thank you very much. It was really eye-opening, like how much is going on in Kazan University, and there is a lot of the progress, especially on the pharmacy side. But also, I think it gives the visibility to the people like us what can be done back home in Pakistan. With that, thank you very much and have a wonderful evening. Enjoy the rest of the weekend, and we will talk very soon. Thank you. Bye-bye.
Thank you.